Hello and welcome to the Wildflower Society's first webinar event. My name is Katie and I will be tonight's host and moderator. I would like to thank you all for taking the time to attend tonight's event, and we really appreciate the overwhelming response we've had so far. I would firstly like to acknowledge the traditional custodians, the Woodjuk people of the Noongar Nation, where we are hosting and recording this webinar. We also acknowledge the traditional custodians of the various lands you are joining from. Tonight's webinar will go for about 45 minutes, and afterwards we will follow up with some Q&A. So please make sure you remember any questions you have throughout the talk and save them for the end. We will do our best to get to everyone, but we do have quite a few people attending tonight, so we will try our best. You can write your questions in the chat box or raise your hand in the toolbar and we can unmute your mic so you can ask yourself. And if you do have any questions about how to set anything up, please send them through now so we can try and help you out before we begin tonight. I would like to introduce tonight's guest speaker, Emeritus Professor Hans Lambers from the School of Biological Sciences at the University of Western Australia. Tonight they will be discussing how dirt-poor soils, pesky parasites and friendly fungi are shaping the greatest plant diversity on severely phosphorus-impoverished soils in the biodiversity hotspot of southwestern Australia. Hans will be diving into why species with a less effective phosphorus acquisition strategy coexist with species far superior in extracting phosphorus from our extremely poor soils, like our Proteaceae species, and unlocking the next chapter on how native parasites contribute to the megadiversity in the southwest. I'm going to pass you over to Hans now, and thank you all for coming, and we hope you enjoy tonight's first webinar.
Thank you very much, Katie. On one hand, I feel honored to be the first person to actually give this talk, this webinar, but at the same time, I must admit I'm also a little bit nervous because both you and I and the Wildflower Society really want this to work. So neither you nor I are allowed to stuff this up tonight. Having said that, let's just get started. I'm going to talk about dirt-poor soils, and you can really see the dirt-poor soils at the left of my slide over here. We've got plenty of them in Western Australia. But I'm also showing you at the bottom over here is a water-mould, an oomycete, such as Phytophthora or Pythium. That's the pesky parasites I want to talk about, because those pesky parasites, they may sound nasty, but they are still very important to maintain the megadiversity that we have here in WA. And then, of course, at the top, you see this toadstool. That's my friendly fungus, and will play a major role in accounting for the fantastic plant diversity that we have here in the southwest of Australia. So let's get started. Well, most of you will know that the southwest of Australia is a global biodiversity hotspot. When Norman Myers, he was actually my neighbor at Utrecht University, where I was a professor before I moved to the UWA. He was a part-time professor there, and we actually had an office next to mine, so we often talked about all this sort of stuff. And when he published this paper in 2000, he only acknowledged 25 biodiversity hotspots, but that by now has actually increased to 36. And you can really see number 12 is our own biodiversity hotspot. But before you start beating yourself on the chest, because we have this biodiversity hotspot, let me tell you what exactly a biodiversity hotspot is. To qualify, an area must have lots of the world's native plant species, 1,500 at the very least. Well, we can tick that box because we have about 8,000 in the southwest. So we really qualify in that way. But we must also have lost 70% or more of the primary vegetation. And of course, when we cleared the wheat belt with the slogan 1,000 acres a year, we lost a lot of the primary vegetation. But that actually still goes on. Banksia woodlands are still being cleared. And I'm particularly interested in the Greater Brixton Street wetlands right now, where the city of Gosnells wants to develop the whole area into an industrial precinct right up to the fence line of Ellison Baird Reserve and all the other reserves that are there. And their claim is that having no buffer is better than the buffer that is expected by the EPA. Absolutely appalling. It's an appalling report. And together with the Wildflower Society and lots of others, we will actually fight against that. So let's first have a look where that biodiversity is. But before I go there, let's look at our major vegetation types. Well, in the far south, in the high rainfall area, we've got the tall forests, the tingle and the karri, of course. And then a bit further towards the west and the north, we've got the Jarrah Merry forests. And then a little bit further inland, we've got the woodlands, and then we've got the Kwongan heaths and the Mallee heaths. And then even further inland, we've got a mosaic of woodlands and shrublands. So that's the vegetation types. But where is our biodiversity? Well, for that, I use a paper by Steve Hopper and Paul Goya, which they published in 2004. And you can really see what jumps out is a number of national parks, like Lezour National Park over here, Stirling Range National Park, and Fitzgerald River National Park. They are our three iconic national parks where we have a tremendous species richness. But it's also true for Greater Perth. And that in Greater Perth, most of that biodiversity is actually kept in only 1% of the total area of Greater Perth. And that happens to be the area including the Greater Brixton Street wetlands right up to Lesmurdy Falls, which we would like to see developed into a regional park. But it's really under threat right now from the proposal that I was referring to by the city of Gosnells. So the richness. But they also looked at endemism, so at species that occur nowhere else on the planet. And again, these same three places pop up. And Greater Perth, again, is also included. So it's an incredibly rich area. So that's where the biodiversity actually is. Now, there's a number of factors that play a major role to account for plant species richness. And first, I would like to mention geological stability. Here we've got some of the volcanoes in Chile, for example, and you can see at the top right what actually happens after the volcano has done its work. It's the bare landscape. Lots of species were actually demolished. So to actually evolve in that area, it's actually pretty tough. So geological stability is incredibly important for species evolution to actually continue. Climatic stability is also important because if you look at this glacier over here, glaciers are absolute bulldozers. They lead to mass extinctions. But the last time we had glaciers in this part of the world, that's actually something like 250 million years ago. So forget about their effects on evolution of the plants as we know right now. And the third point I'd like to make, and I really get back to that because Katie mentioned that already, the soil infertility is incredibly important. And we have a lot of what I should refer to as severely phosphorus-impoverished soils, but that's a long word. So I'd like to refer to it tonight as crappy soils. And I think the message gets across when I talk about crappy soils. We've got lots of them, and don't knock them because that is where we actually have our greatest biodiversity. So it's all about Mother Nature on one hand, but it's also about Father Time. And when they get married, well, evolution actually continues. Now, biodiversity, of course, on one hand means that we've got lots of species, but for a plant scientist who wants to understand what's actually going on, it's also about patterns and processes. So you can admire and enjoy them, that's fine, but I would like to understand the patterns and processes that actually underpin that tremendous biodiversity that we have over here. And this is a graph that I used in a book that I edited, Plant Life on the Sand Plains in Southwestern Australia. And that clearly shows a pattern that we see in the Southwest. And that pattern is illustrated, first of all, by this line at the top. So species richness is plotted along the y-axis, and then total soil phosphorus is plotted on the x-axis. And what you can really see is the biodiversity increases when the soil phosphorus concentration declines. So the crappier the soils, the richer the flora. And what I would also like to point out is this red group over here, and that red group refers to Proteaceae. Those Proteaceae don't really like those richer soils, but they are very abundant on our crappiest soils. That includes Banksias, Grevilleas, Hakeas, Isopogons, and you name it. They absolutely like those nutrient-poor soils. They don't like the richer soils because they're actually, a lot of them are actually very phosphorus sensitive. I always say they are so phosphorus sensitive. If you want to use the word superphosphate when standing next to a Banksia, make sure you whisper because they are so sensitive that might actually kill them. So there's a message over here, and that message is, if you want to conserve biodiversity, or if you want to create it in your own backyard, then you must keep the soil phosphorus levels at an absolute minimum. Now, that's a message I also try to get across to DBCA, because DBCA loves to spray phosphite to combat Phytophthora. And phosphite in soil actually gets converted into phosphates. So they are continually enriching the soil with phosphates. And they do that at a rate very similar to what wheat belt farmers use. But the wheat belt farmers, they export that wheat when they harvest the grain. DBCA does not harvest anything. So that phosphorus actually accumulates in the soil. So they will affect the soil fertility, the soil phosphorus. Now they tell me, oh, it's only a stopgap measure while we find something else. But they have been telling that since the last century, and they still use it today. And if they were looking for an alternative, then you can say, yes, it is a stopgap measure, but there's no action when it comes to that in DBCA. Nobody is actually looking for an alternative to phosphite. But I think it's urgently needed because you will affect the biodiversity if you keep spraying phosphite. Now, why do I drone on about phosphorus? Well, it is actually because phosphorus is essential for all life on Earth. Whether you are an animal, a plant, a fungus, or a bacteria, or if you are a virus like a coronavirus, they all depend crucially on phosphorus. Without that, they cannot function. When it comes to us, phosphorus is literally in our bones, but it is also in our DNA. Or if you are a virus, it may be in your RNA, but none of these organisms can actually live without phosphorus. Now, I often get asked the question, well, look at the periodic table. There's so many elements. Why can't you replace that phosphorus by one of these other elements? Well, the closest element to phosphorus is actually arsenic. And I'm not sure whether you're very familiar with arsenic, whether you have read some of Agatha Christie's books, but Agatha Christie books always likes to knock off a few of the people featuring in her books. And she uses arsenic because arsenic is the poison of Agatha Christie. And at the right, you see Napoleon. And Napoleon is not very happy over here. On his deathbed, he was actually inhaling arsenic. And that arsenic basically evaporated from the wallpaper they like to use in those days. And that basically shows you that arsenic compounds are extremely unstable. That's why it evaporated, and that's why Napoleon was actually inhaling it. And they found the arsenic in his hair. And the French accused the English of poisoning him. Well, they weren't actually poisoning him deliberately, but he was poisoning him, that wallpaper, if you like. So replacing phosphorus by arsenic only creates very unstable compounds. And there's no way that any plant, animal, bacteria or fungus could actually survive without phosphorus. Now, when you go to the rest of the world, and that's the story I told my students when I was at Utrecht University, then we find lots of association between plants And here you see a plant root. This is actually a cereal. And what you see is that the cereal was associated with fungi. These are mycorrhizal fungi. So this is a true symbiosis between a fungus on one hand, a mycorrhizal fungus, and in this case, a cereal. And you can really see what these fungi are doing. They can pick up phosphorus that is far away from the root surface over here. Bear in mind, phosphorus does not move in soil. It gets stuck to all sorts of soil particles. It doesn't move. So if you want to get it, well, you've got to scavenge for it. And you can really see that this is exactly what these mycorrhizal fungi are doing. So these mycorrhizal fungi, they pick up the phosphorus for the plant, and in exchange for that, they get provided with the bread and butter by the plants. Now, don't get me wrong. There is also plenty of mycorrhizals in Western Australia, but I hasten to add that symbiosis is not really as common as it is in the rest of the world, and that is odd. That really surprised me when I moved to WA in 1998. I knew that the soils were poor. I didn't quite know how poor because we didn't have all the data then, but I knew they were poor, and I also knew that there were lots of non-mycorrhizal species, and that was really puzzling, and you could ask anybody, and they couldn't really tell you the answer. I'm going to tell you the answer in the next few minutes or so. So this is what I was just talking about, reported in a paper by Mark Brundrett. So all these segments that we see pulled out over here, they are all groups of plants that have mycorrhizas. Orchidaceous mycorrhizas, for example, and here are the arbuscular mycorrhizas that I showed you in my previous slide. And you can see that globally, only 18% of all species in the world cannot form ever an association with a mycorrhizal fungus. 18% for the whole world. Now let's go to WA. Then we see that 40% of all species there are non-mycorrhizal. And that includes all the species like the Proteaceae, all the Banksias, Hakeas, and what have you, all the sedges, all the restios, and a whole bunch of other groups of plants as well. I will talk about them later on. So that was really puzzling. And when I moved here in 1998, as I mentioned, nobody could actually tell me why there are so many non-mycorrhizal species. And the answer is actually quite simple if you have the wisdom of hindsight. But in 1998, I did not have the wisdom of hindsight. And so it took a while before it all sank in. Now, don't get me wrong. There are lots of groups of species that are mycorrhizal. Here are a few examples of arbuscular mycorrhizal species, for example. You're all very familiar with them. Here are examples of ectomycorrhizal species, very common in the Proteaceae. Here we see examples of erigoid mycorrhizas in the Ericaceae, of course. And here we have examples of the orchidaceous mycorrhizal species in the orchids, of course. But that's not what I want to talk about. I want to talk about that large group, 40% of our flora, that is non-mycorrhizal. And I don't include all the families here, just a few of the most important ones, the Proteaceae, the Hemoduraceae, and of course also the coniferous plants, of which we have more in the Southwest than anywhere in the world. And so the question then is, why are there so many non-mycorrhizal species in the Southwest biodiversity hotspots in a large range of families? And of course, the question really is, how do they get their phosphorus if they don't get any help from mycorrhizal fungi? Well, this is what they do. They produce these weird and wonderful cluster roots in this Hakea species, for example. And this is clean because we've grown this plant in nutrient solution. But if you dig them up from a fire break in the Zoo National Park, then they look like this. And for a Banksia, they look like this if you grow them in nutrient solution. And if you have them in a pot, they look like this. Compound cluster roots. And in sedges, we have these so-called dorsiform roots. Not all sedges have them, but many have them. And if you dig them up, they look something like this. Sand-binding roots, typical for most of the Biodiversity. Again, they play a very important role, and they do something very similar to what I'm going to talk about for the sedges, or the Proteaceae rather. But before I go and dig down how they actually get their phosphorus, I'd like to talk about our Durian Bay chronal sequence. When we go back about 140 million years ago, what we are looking at right now, that is exactly where India was attached to Australia. And then when India broke away and moved north at an incredible speed, bumped into Southeast Asia, gave rise to the Himalayas and the Tibetan Plateau, then that big gaping hole was filled with beach sands and sediments that eroded away from the old plateau. And so we have a series of dunes that were formed over thousands of years, millions of years actually. And the reason why we have a series of dunes, that is because the level of the sea went up and down and up and down. And that's why we ended up with 3 dune systems. The youngest, only a few years old, up to 7,000 years old. The next one starts at 120,000 years, up to 500,000 years old. And the oldest dunes, that's the Bassendean dunes, they are about 2 million years old. And so here we've got those 3 dune systems. The quintuple dune systems, they begin like this, and in stage 2 and in stage 3, we gradually increase in the vegetation, but also a change in the species that actually occur there. And then we go to the spearwood dunes, much more species rich, a lot more Proteaceae are coming in. And then finally we go to stage 5, the Bassendean dunes over here. Then the Proteaceae really rule, as I will explain in a minute. Now, what is typical for soils that got older and older, what is typical is that they start with very high phosphorus concentrations, so close to the ocean. Think of City Beach or Cottesloe or whatever you want to think of. That is where, on those dunes, we have our richest soils. Imagine beach sands, our richest soils. And then when soils get older, here to 7,000 years old, here to 120,000, here to 2 million years old, then we can really see that phosphorus gradually disappears. So our soils are severely phosphorus-impoverished. Our soils are very crappy for the simple reason that they are very old. Of course, they never started with very high concentrations over here. Beach sand is not particularly rich. It may be rich in Western Australia, but not rich by world standards. But we lose that phosphorus over thousands and millions of years. And so that's why we end up with these incredibly nutrient-poor soils. Nitrogen is a very different story because the beach sand doesn't contain any nitrogen. All of that will have to be brought in by microorganisms that are capable of fixing nitrogen. And then the nitrogen also goes down again. And the reason for that is simply you cannot fix nitrogen unless you have phosphorus. And if the phosphorus disappears from the system, you stop making nitrogen. Nitrogen disappears from the system, partly because of fires, partly because it leaches out. And so the nitrogen concentration also goes down. So this is that first stage. High in phosphorus, very low in nitrogen. And the vegetation is actually, well, pretty sparse, with a lot of bare soil. A few hundred to 1,000 years older, things have really changed. Now we have a full cover. There's plenty of phosphorus, and actually in the topsoil, there's more phosphorus than there was before because the plants have actually taken that up from the subsoil and their litter is dumped on the top. So that's why the phosphorus concentration actually goes up. And you can also see that the nitrogen concentration has really gone up because all the nitrogen fixers, acacias, alocasiarinas, and what have you, they've all done their job and fixed a lot of nitrogen. But then after 7,000 years, we can see that the phosphorus level already is going down and the nitrogen level is also going down, and gradually Proteaceae move in. And then we go to the dunes that are more than 120,000 years old. Now we see lots of Proteaceae. We've lost most of the phosphorus and we've lost a lot of the nitrogen as well. And then we go to the oldest dunes. then we have lost just about all the phosphorus that was ever there, 2 million years old, and we have lost a significant amount of the nitrogen as well. But it is an incredibly rich flora. This is where our greatest biodiversity is. Now, there are many chronosequences all over the world. And this gentleman over here is one of my heroes, and he worked on the chronosequence in Hawaii. The chronosequence there was not the result of beach sand being leached and eroding away. The chronosequence there is the effect of volcanic activity. And then the islands that were produced by volcanic activity basically lost their phosphorus as the older they got. So he did a lot of pioneering work on the chronosequence in Hawaii. And he visited our chronosequence in Bay. And when I visited, when I took them there together with his wife, they were like kids in a candy store, because every few meters you found a new species, and then you walk 10 meters and another new species and another one, lots of carnivorous plants, lots of orchids. He was absolutely rapt. And he said, well, when you visit Hawaii, it only will take me about 20 minutes to explain to you all the species that are there, because there's only a handful of species there. And of course, here we've got hundreds of species. So no wonder he was very keen to visit our chronosequence rather than the boring chronosequence in Hawaii. So let me focus on two families that are dominant, of course, in Western Australia. The Proteaceae on one hand, like eucalypts, and on the other hand, we have Proteaceae. That's the Banksias. And of course, being mycorrhizal species, these plants, they prefer the richer soils over there. And of course, these Proteaceae, I pointed out before, they really love those really nutrient-poor soils. But the question is, how do they do that? And I already pointed out, they make these weird and wonderful cluster roots, but how do they actually work? How do those cluster roots get phosphorus out of the soil that those mycorrhizal plants like eucalyptus actually fail abysmally? Well, we studied that in great detail, and we looked at the development of these cluster roots. And what you can really see is these cluster roots, they only live for about 3 weeks. That's incredibly short for a root. Some live 3 months, some may even live more than three years. But these only live for 3 weeks. And if you only live for 3 weeks, that basically means that you are in incredible hurry, and you have to work very hard to actually make that cluster. And if you work very hard, it's just like when we run very fast, well, you've got to respire very fast. And that is exactly what these roots do. So when they are young and are rapidly growing, they've got very rapid respiration rates. And then when they are maturing, their respiration rates actually goes down. So first they use a lot of their sugars to respire and build cluster roots. And what do they do then with all the sugars that are imported from the leaves? Well, they produce and they release what we call carboxylates. And my next slide will actually explain what carboxylates actually are. But if you like, think of them as acids, like malic acids and citric acid, for example. It's not quite right, but I'll explain in a minute what it is really all about. So these are carboxylates. And these are actually they are actually part of what we call organic acids. And here are 3 examples of organic acids that are commonly released by plants like the Proteaceae and the Cyperaceae. But they are not actually acids, they are actually the anion component of the organic acids. And the main one in Hakea, for example, is citric acid. Other plants, they prefer to release malic acid or oxalic acid, but they all basically do exactly the same. But I should point out, it's not actually the acid that does the trick. It's actually the anion component of the molecule, and that's what we call the carboxylate. Well, that's a lot of science. Let me go to this graph over here and explain why it's not acids that really do the trick. Imagine we go to the youngest soils here in close to the beach, for example. They are alkaline, they are full of chalk, they've got a very high pH, and you can see looking at the width of the band referring to phosphorus, that is where phosphorus is actually poorly available. So here, if you acidify the soil, you actually make phosphorus more available. So that makes perfect sense. But it only makes perfect sense on those young dunes where there's plenty of phosphorus. But if you go to the older dunes, like the spearwood dunes and the Bassadine dunes, there we are dealing with an acid pH around 6 or even lower than 6. And there, if you make your soils a little bit more acidic, well, you may actually decrease the availability of phosphorus. It's a simplification of the real world, as Jim Barrow will be happy to point out, but it suffices for the point I'm trying to make over here, that simplification is not really the important trick these plants actually play. It's the release of the carboxylates. So these are the soils we are really dealing with, and they are slightly acidic, so making it more acidic is not a good idea at all. Jim Barrow gave me this slide, and you're probably all familiar with Jim Barrow because he's one of your distinguished members, but he also was, and still is, a plant scientist, a soil scientist, I should say, and he says soils actually look a little bit like that. Now, a couple of years ago, Jim told me he was NBN, and I thought I knew what NBN meant, but I didn't quite know how that related to Jim's status. And he explained to me that NBN meant nearly bloody 90. Well, that was a couple of years ago, so he must be OBN by now because he is already over bloody 90. But he's still active and very sharp. And he tells me that soils really function like this. So we have soil particles, these brown blobs over here, and then we have phosphorus molecules, phosphorus ions. That's these yellow dots over here. And some of these yellow dots are in the soil solution. And those are the phosphorus molecules that plants or fungi can actually take. But in our soils, in very nutrient-poor soils, most of that phosphorus is actually glued to these soil particles. And that phosphorus, most plants cannot actually take up because it's not in solution. So how do you get that? Well, that's where the Proteaceae come in. Because what they really do is they produce these cluster roots. And these cluster roots, as I pointed out, they release all these carboxylates. And these carboxylates, they manage to mobilize the phosphorus that is in the soil in sand, but they can even get some of the phosphorus out of this gravelly stuff, of which we have a lot in some of our soils over here. So really, the system all works. And for my international audience, I really like to give all the details and tell that the carboxylates are released from the roots over here into the soil over here. And in the soil, they replace the phosphorus because they are negatively charged, and phosphorus is also negatively charged. And they push the phosphorus in solution, and the plants then take it up. So this is basically what I call a mining strategy. They don't actually scavenge for phosphorus like the mycorrhizals do, and they can pick up phosphorus that is far away from the root surface, but these actually mine the phosphorus that is tightly bound to the soil particles or in the gravel, as we see over here. So that's basically the big trick that Proteaceae, Cyperaceae, Haemodraceae, Restionaceae, and other species with these specialized roots actually do. And now I made a mistake because I went to the last slide. My apologies. So that was the scheme I tried to explain, but let me quickly summarize this part of my talk. When it comes to phosphorus acquisition in plants that release these carboxylates, like in this Hakea over here, they've got specialized roots. And what these specialized roots really do is they vast amounts of carboxylates, such as citrate and malate. And what's really important is citrate and malate, they mobilize the phosphorus that is bound to soil particles. And that's really a mining strategy. And that's what they do, something that mycorrhizas fail to do. So going back to my Proteaceae over here, well, they associate with mycorrhizal fungi, and they are great scavengers. They can pick up phosphorus that is far away from the root surface. That's on account of their association with mycorrhizal fungi. But then here we've got the miners, and the miners, they have their cluster roots, and they can pick up phosphorus that is not available for these plants, like they have association with mycorrhizal fungi over here. But there's one thing of this graph which isn't quite right. So we've got the scavengers at the far right, we've got the miners at the far left, and then we see some of the Proteaceae that share a habitat with the Proteaceae, although they presumably do not have a mining strategy. So how does that work? Well, that could mean that my ID is not quite right. It can also mean that it's a bit more complicated than the simple story about miners and scavengers I was talking about before. And that is indeed the case. So here we've got our Banksia with cluster roots. Here we've got one of our Proteaceae, a mycorrhizal species, and we decided to grow them either alone in monoculture or together in a mixed culture of the two. So here is my Banksia sharing the root environment with the Schultzia over here, and we actually clearly showed that if you grow them together, the Schultzia actually grows a lot better. So the cartoon that actually explains this is over here. So here we've got my Banksia with its weird and wonderful cluster roots, and they release a lot of carboxylates and they mobilize a lot of phosphorus. And what this and other Proteaceae actually do is they snuggle up to a plant that does all the hard work, and they basically take up some of the phosphorus mobilized by this hard-working Banksia species over there. So they are what we call facilitated by this species that is very effective and very efficient at mobilizing the phosphorus, not just for itself, but also for its neighbor. So it facilitates the phosphorus uptake of its neighbor. And so that accounts for the fact that you still have a lot of mycorrhizal species in an environment where mycorrhizas are not actually good enough to take up all the phosphorus. But wait a moment. Nobody can be good at everything. These Banksia may be very good at taking up phosphorus, but we suspected that they might also be very sensitive to these pesky parasites illustrated over here, these oomycetes like Phytophthora and Pythium. And I'm not talking about Phytophthora cinnamomi, which is an introduced pathogen. I'm talking about native pathogens which you can find all over the place in the southwest of WA. And we thought that these short-lived roots, which are very good at getting phosphorus, they are perhaps very susceptible to these root pathogens, these oomycetes, these water-moulds shown in my top right corner over there. And well, you can read all this text, you can look at the graphs, but one picture is worth more than 1,000 words. So let's look at these two pictures over here. That's 2,000 words. At the left, in A, we've got Banksia leptophila, which is grown in its own soil, collected from the Julian Bay area, where this species is native. But because I'm a plant scientist and I'd like to grow my plants in healthy soil, I first pasteurize this soil over here. And you see this plant grows really very well. In B, we grow the same species, grown in the same soil, collected from its natural habitat in Julian Bay, but now we did not pasteurize this. And pasteurization, of course, kills all the pathogens. And here, when we've got lots of pathogens, we can really see that this plant is really hammered. It's not killed by the pathogens, but you can really see its growth is severely reduced by the pathogen, by the oomycete pathogens. And in this case, Pythium irregulare was one of the key oomycete species, but quite often we also find a lot of Phytophthora species. So the cartoon really is not as simple as I talked about before, when we had no pathogens and we had a non-mycorrhizal plant with cluster roots over here and a mycorrhizal plant with no cluster roots, and this plant facilitates the uptake of its neighbor. So there's clearly some facilitation, but there's also strong competition because the phosphorus that is mobilized is closest to the roots of this species, so that will have first pick in taking up the phosphorus. Now, the real situation, the real world, and that is the situation with pathogens. And we know that the pathogens really affect the non-mycorrhizal plant with cluster roots. We saw that in the previous slide. But the other plant, which has mycorrhizas, really benefits from that. And why is that? Well, that is because mycorrhizas are not only good to take up phosphorus when the soil conditions are right, but mycorrhizal fungi, they are also known, very well known, to boost the defense against pathogens in its tissues. So that is why this plant can actually grow despite those pathogens, because it is associated with mycorrhizal fungi and they provide the defense against these pathogens. But my poor Banksia doesn't have that defense, and so it's hammered by the pathogens. So that's why my friendly mycorrhizal fungi come in. They may be inferior when it comes to phosphorus uptake, but they protect the mycorrhizal hosts like the Proteaceae, those pesky oomycete pathogens I was talking about. So together, a very efficient plant when it comes to phosphorus uptake, but sensitive, susceptible to oomycetes. A mycorrhizal plant that is not very good at taking up phosphorus, but is defended against oomycetes, against the pathogens, because it is mycorrhizal. Together, those four players account for this tremendous biodiversity that we have. So let me summarize this part of my story. Why do we have this tremendous biodiversity in the Southwest? Well, as I said, the greatest biodiversity occurs on our poorest soils. And on those very poor soils, we've got those plants that are very good at taking up the phosphorus, and they facilitate the phosphorus uptake of their neighbors. And facilitation is far more important than competition in this biodiverse, in this megadiverse habitat over here. So the Proteaceae are very efficient at getting phosphorus, but you can't be good at everything. They are also very susceptible to pathogens. And the mycorrhizal plants, well, the mycorrhizal plants, they may be less effective at getting phosphorus, but they cope a lot better with the pathogens, courtesy of their association with mycorrhizal fungi. So it's these interactions among the four players that account for our megadiversity in the Southwest. Now, I've got two questions to finish off my talk. What would happen if the oomycete pressure became too high? So in my example, they didn't actually kill the plants, but if the oomycete pressure became greater, would they actually kill some of those plants? And what would happen then with the biodiversity? And then, one of my first slides, borrowed from Steve Hopper's work, showed that the tall forests, they are not nearly as diverse as our sandplain flora. So why is that? So let me first start with this question. If the oomycete pressure became too high. Now, luckily, there are a range of cronus sequences in WA. I talked about our Jurien Bay cronus sequence, but we have three others. And at the other extreme, we have our Warren cronus sequence. And in Jurien Bay, we've got an annual rainfall of 433 millimeters. And of course, on the south coast, along the Warren cronus sequence, we've got 1185 millimeters. So a lot wetter, favoring oomycetes. They're water-moulds. So here we see the oldest dunes along the Jurien Bay cronus sequence. And here we see the dune system of the same age, but here at Warren, where the rainfall is much higher. We've got a higher rainfall, we've got a lot more litter, and these are elements that actually favor oomycetes. Well, in Jurien Bay, we've got dozens of Proteaceae. I think there's probably about 100 or so. Adenanthus, Banksia, you name it. And as I said, it's a typically non-mycorrhizal family. And in Warren, we have only very few Proteaceae. We've seen 6 Banksia species, there's 2 Hakea species, and there's only 1 Personia species, and that's all. So it's only 9, as opposed to more than 90 Proteaceae species in Warren compared with Jurien. And the soil moisture and the litter, they would really favor oomycetes. We are actually following that up, doing the work right now and actually quantifying that. And our hypothesis is that those oomycetes, they likely wiped out most of the Proteaceae, with the exception of the 9 that I've listed over here. And what is really interesting is, of those Proteaceae, of those 9 that we have studied at Warren, all are colonized by mycorrhizal fungi, illustrated over here. So here is this Banksia seminuda, and if you look at their roots, they have the typical mycorrhizal structures that you do not find in Proteaceae normally. Here we've got a Hakea species. Again, you do find mycorrhizal structures that are not typically found in Hakea species at all. We looked at a number of other species as well, and all the ones we looked at in Warren, you can all find mycorrhizal structures over there. When you go to Jurien Bay or elsewhere in Australia, you will usually not find any mycorrhizal structures in Proteaceae. So the role of mycorrhizal fungi in the Proteaceae would then be, well, they must be irrelevant for P acquisition, because the soil phosphorus availability is way too low for them to be effective. And we believe, and we are going to test that with glasshouse experiments. One of the postdocs is working on it, and 2 of PhD students are also working in this space. The hypothesis is that those mycorrhizas in those Proteaceae in Jurien Bay, and not in Jurien Bay, but along the Warren-Kroner sequence, they are important to boost the defense against oomycetes. So those ones that associated with mycorrhizal fungi again, those can actually make it along the Warren-Kroner sequence. And all the others that did not discover that trick, well, they can't actually make it over there. And then my last question, why are some of our tall forests not as diverse as our sandplain flora? You're all familiar with the Kerry forests. Kerry forests are majestic, but they are not particularly diverse. We started work on the forest shown at the left, that's Yarry, Eucalyptus patens over here. And the thinking in the beginning was perhaps these soils where Kerry and Yarry, or blackbutt, if you like, where they actually grow, perhaps those soils are a lot richer, and perhaps that accounts for their lower biodiversity. Well, is that really true? Are soils supporting Kerry and Yarry, are they really richer in phosphorus? Well, let's go back to Sir James Mitchell. Mukau Mitchell, he was usually called in the press, because he had a wonderful scheme, a settlement scheme in Margaret River. And at the left, we see a wonderful postcard he used to send to the UK to lure over people to come to WA, because it was such a wonderful environment with good rainfall, with very rich soils, and you could really start your dairy farm over there. Well, it was a complete failure. And it was a complete failure because the soils were deficient in lots of nutrients, and the phosphorus that was there is so strongly bound that no crop and pasture plants were actually capable of taking up phosphorus from those soils. So it was an absolute failure. So the soils are not richer where Kerry and Yarry actually grow. But what would happen if these mycorrhizal plants like Kerry and Yarry, when they would actually manage to discover the trick that I showed you is so successful in Proteaceae and also in Cyperaceae? Is that possible? Well, how do we test that? So again, I pointed out that Proteaceae have these cluster roots. Well, Myrtaceae don't. Carry doesn't, and Yarry doesn't either. But they could still release carboxylates. That's possible. And these carboxylates, as I pointed out, they mobilize phosphorus, but they also mobilize manganese. And the manganese uptake in plants is actually very poorly controlled. So if they mobilize manganese in their environment, they will actually take up that manganese. So there we go. They mobilize the manganese and they take up the manganese. And so I can actually measure the manganese concentration, and that tells me what these plants were actually doing below ground. And so we went to a site where Yarry grows, and we sampled a range of species there. On one hand, we sampled a Banksia grandis that occurs in the Yarry forests. And in the Yarry forest, the Banksia grandis has very high manganese concentration. You would expect that because we know that these cluster roots release massive amounts of carboxylates. They mobilize the phosphorus, yes, but they also mobilize the manganese, and that is then taken up. On the other extreme, we take a grass tree. A grass tree is mycorrhizal, but basically takes up its phosphorus immediately after a fire when it's one of the first to actually regrow. That's when it capitalizes extra phosphorus in soil after the fire. And it always has low leaf manganese concentration. So that's our negative reference, if you like, and our Banksia is a positive reference. And then our targeted species is Yarry. And we can really see it has leaf manganese concentration somewhere in between my positive and my negative reference. So that suggests that it must be releasing carboxylates. It's not as effective as Banksia because the manganese concentration is not quite as high, but it does a lot better than my grass tree over there. That suggests that you need to follow that up. So we dug up some seedlings from that property near Varuna. We grew them in the glasshouse, and lo and behold, when you measure their exudation rates, these plants release indeed carboxylates. Not citrate and malate like the Banksia does, but oxalate. But oxalate does exactly the same in soil as malate and citrate. And then we thought, well, let's look at that range of other species that occur at the Yarry sites. And indeed, we measured the exudation rate of a range of species that all had high leaf manganese concentration. And we can really see all these species, Banksia, Corymbia, you name it, they all release carboxylates, and so does Carry. And now we're on to something really very important. Carry and Yarry, they clearly have traits like carboxylate-releasing phosphorus-mobilizing Proteaceae. Okay, they are not as good at it because they don't build up the manganese concentration to the high level, telling us they don't release carboxylates as effectively or not as abundantly as the Proteaceae, but they do release them, and so they do have a good strategy to get the phosphorus from those soils. But being Myrtaceae, they're also mycorrhizal, so they are also very well defended against plant pathogens. So they don't need to be facilitated. They don't need to depend on a Banksia or a sedge to actually get their phosphorus. They can do it all by themselves. So we can expect them to be very against pathogens, and they know how to get the phosphorus from very nutrient poor soils. They basically have the best of both worlds, but that basically means that the biodiversity is gone. Facilitation no longer plays a role, at least not to the same extent as on the sand plains, and that's why they confiscate the whole space and outcompete a lot of other species over there. And now you can see why Mukau-Mitchell's settlement scheme was a complete failure. But of course, we're talking about 100 years ago. 100 years ago, politicians did not listen to scientists. Aren't we lucky that we live in a completely different world? In the 20s, we had Patterson, the first professor of agriculture at UWA, and he warned the politicians against salinity. But the politicians basically told them to go away. They did not want academia. They did not want academics to stand in the way of progress. And the same idea existed in 1924 when it came to the Mukau settlement scheme. 100 years later, fortunately, we are in a completely different situation. If we now talk about climate change, the politician asks, what should I do to act as quickly as possible? How should I decarbonize the economy? And what should I not do? There is a role for science underpinning political decisions. It was a complete stuff-up 100 years ago, but have we really moved on a great deal? I'm not entirely sure. Well, I really would like to acknowledge that I was involved in all this work and all these papers that I cited, but I was actually dependent on a host of collaborators at UWA. A lot of students were involved, a lot of international and local and Australian collaborators were involved, and it's only because of that collaboration that I was able to give this talk today. Thank you very much for your attention.
Thanks, Hans. That was really, really fascinating. If anybody has any questions, please make sure you send them in or raise your hand.
Well, while you think of your questions, I'm going to pour myself a glass of wine because I think I've deserved that.
Very well deserved.
There's questions popping up in the chat box.
So we have a question from Mike, and they've asked, how does Jarrah fit into the picture?
That's a fascinating question. Jarrah also releases carboxylates, but unlike a lot of other eucalypts we have looked at, it stops doing that when you give it more phosphorus. So Yarry and Kerry, they always release carboxylates, and you can give them more phosphorus and they would still continue releasing all these carboxylates. Jarrah does not do that. And that explains why on the UWA campus and elsewhere in the suburbs, you see that these Jarrah plants, they are in very poor shape because in the western suburbs, people irrigate with alkaline bore water. And that alkaline bore water makes manganese and iron very poorly available. So they run into iron or manganese deficiency. That wouldn't be a problem if they release these carboxylates because they would actually balance for that. And if you look at Mary does not suffer from the same problems as Jarrah when you irrigate it with alkaline bore water. And that is because Mary releases carboxylates, but it does that irrespective of how much phosphorus you give it. Whereas Jarrah, when you give it a little bit of phosphorus, then it will actually stop doing that and makes it impossible to get all the micronutrients such as iron and manganese. And on the UWA campus, I spread iron sulfate around to actually acidify the soils to counteract that. For the Marys, I don't need to do that, but for the Jarrah, I do need to do that. Because the problem on the UWA campus is that they not only irrigate with alkaline bore water, but they would also have, over the years, they would also soil with phosphorus, of course. So Jarrah is a different story from what Mary and Yarry actually does, or Kerry for that matter.
We have another question from Jackie who asked, are there any alternatives to using phosphates to treat Phytophthora, and how do we encourage DBCA to find an alternative?
I've talked to people at DBCA, and I actually was very keen to develop an ASC linkage project, which basically means that DBCA would have to put some money on the table, but that would only be something like $30,000 a year. And then the rest of the money would come from the Australian Research Council. And we would be doing the research jointly to look for alternatives. And the idea is these days we've got a lot of metabolomics, transcriptomics, genomics techniques, but that will actually tell you what phosphite actually does. And phosphite basically makes the plant defend itself better. So if you would actually look at all the molecules that pop up and you spray it with phosphite, then that will may actually give you a clue as to what you would really need to boost the defense in those plants. And so once you know which molecules pop up, you can look at other molecules you can spray with that are innocent, that will actually have the same effect as phosphites. So a project like that, I think, could really have a tremendous effect.
Yeah, it definitely sounds like it. They need to definitely review the processes, it seems.
I think they should, because of course, there's also the effect that phosphite affects oomycetes. And of course, I was also talking about the effect of oomycetes in maintaining our biodiversity. So that phosphite that you spray to kill the Phytophthora cinnamomi would also have an effect on the oomycetes that are actually one of the pillars of our biodiversity. So there is every reason to start looking for an alternative for phosphites. And you can't keep telling people that it's just a stopgap measure. They've been saying that since the end of last century, but a stopgap measure is what you use for a short period. You don't use that for decades. So it's really absolutely essential that they look for an alternative. And they can do that jointly with the universities. And I'm sure you could work towards a solution.
Yeah, definitely. We have a question from Kirsty who asked, have you looked at any other fungal or even bacterial species associated with the various plant families? Good ones, not pathogens.
There is a Chinese student who just joined my group who is very keen to look at those friendly bacteria that do things together with mycorrhizas, for example, because we can see that the mycorrhizal fungi, they boost the defense, but she actually wants to know whether there are associated with the mycorrhizal fungi that perhaps help in that whole process. So that's the sort of project that she is thinking of, and she is just writing her proposal right now. So yeah, we are exploring that. That would be a new area for me, but hey, why not? Got a few more years to go.
While we wait for any more questions, there is a short survey at the end of the webinar as well. So when you do leave, if you do have any time, please fill it out. We would really appreciate and value any feedback that you have.
And if the feedback is positive, we may make this a regular event. Not me, but other speakers.
Yes, we are planning on having monthly webinars. So we are hoping to announce next month's webinar and their guest speaker next week. Yeah, Corma asked if we could get a recording of tonight. Yes, it has been recorded, and we'll be we can upload that probably at the beginning of next week, and we'll put it on Facebook so everyone can see it as well.
I agree with Jackie. I hope one day politicians start listening. And Colma, if you can hear me, I moved my walk from Sunday to Saturday, so we can actually have our Zoom meeting on Sunday. I would be available.
We have another question from Faz. They said, thank you for your amazing talk. Have you noticed different combinations of facilitative relationships with other non-mycorrhizal families and other plants?
We are actually looking at facilitation. We just published a paper in the New Phytologist promoting leaf manganese as a tool to actually quantify facilitation. In Ellison Baird Reserve, one of my students did a wonderful project on Acacia pulchella. Acacia pulchella pops up after a fire and it germinates massively and it grows abundantly, and then you see massive deaths a few years later. So it clearly benefits from the extra phosphorus that is available immediately after the fire, and then when it runs out of phosphorus, a lot of these plants actually die. Now, not all of them die. Some of them actually do quite well. And what she actually did is she measured the leaf manganese concentration in Acacia pulchella as a function of distance to a big Banksia. And what you really found is close to a Banksia, Acacia pulchella has high leaf manganese concentrations. So there it was clearly facilitated. And 10 meters away, they were in poor shape and the leaf manganese concentration were low. It was too far away from a Banksia to be facilitated. So that Acacia, and we also found that for Bossia, and we also found it for Hebersia. There we clearly have evidence that they were facilitated mainly by big Banksias. But I don't think it's just Banksias. If you go to Agonis flexuosa, peppermint, for example, along our Warren-Cronin sequence, I think it's probably mainly facilitated by the understory sedges and restios that you have over there rather than the Banksias. So I think facilitation in our environment has been overlooked, but I think it's going to be very important, and we are discovering more and more how important that actually is. And we can use the leaf manganese concentration as a tool to actually quantify that.
While we wait for any more questions, did you want to just give a short bit of information about the Greater Brixton wetlands and the submission session that's on Monday?
Colma may want to make a few comments about that. So the city of Gosnells wants to develop all the area around the Greater Brixton Street wetlands. And we don't object to that, but we believe the Greater Brixton Street wetlands, they really have to protect it because, as I said, in 1% of the Perth metropolitan area, we have about half of all the species that occur there. And it's because of that area that Perth can be considered the most species-rich, the most biodiverse city in the world. So it is, and Ellison Baird Reserve, for example, a reserve in the Greater Brixton Street wetlands owned by UWA, and I do a lot of my research over there, is by all accounts the most biodiverse reserve on the entire Swan Coastal Plain. So this is a very precious area of global significance. So we have the responsibility to actually look after that. The city of Gosnells on the website tell everybody how proud they are on their, if you like, Greater Brixton Street wetlands. But with being proud comes a responsibility. And rather than be driven by greed, I think they should take on board that they have a responsibility to be custodians of the Greater Brixton Street wetlands and to actually want to develop the area right up to the fence of those Greater Brixton Street wetlands is absolutely irresponsible. EPA has very clear rules when it comes to buffer zones around sensitive areas. The more sensitive, the bigger the buffer zone has to be. This report written by this consulting company, Emerge, basically argues in a very qualitative way, without using any science, without any quantitative information, basically argue that having no buffers is safer. It's safer in terms of protecting the Greater Brixton Street wetlands than having a buffer zone of 50 or 100 meters or more. It's ludicrous. There is no science underpinning all of this. And if we know that these Greater Brixton Street wetlands are not just fed by rainwater and surface flow, but to a large extent by subsurface flow, a lot of our wetlands in Perth are actually fed by groundwater, and the Greater Brixton Street wetlands is definitely no exception. If you know all of that, then interfering with that groundwater flow basically means that all species and threatened ecological communities in the Greater Brixton Street wetlands will be under threat. And you are likely going to lose some of the rare species that occur nowhere else in the world, only in the Greater Brixton Street wetlands, if you allow this development to go ahead in the way proposed in this report provided by Emerge. It's an absolutely shonky report. It's shocking what they dare put on paper. And Koma may want to add to this, because she is the mover and shaker behind a workshop that we have, I think, on Monday.
Yeah, I think Urban Bush Care are having a Zoom or an in-person submission session to put together submissions for the Greater Brixton Wetlands. So if you are interested, please make sure you get in touch about it.
So if Kolma is still here, she may want to join us for.
Thank you. I think just quickly, yes. Thanks, Hans. It's on Monday evening from 6:30 to 8 o'clock in the City West Lottery House, and if I put it in the chat, but we can send it out hopefully to anyone that's hooked in tonight. And people like Hans and Kate Taus and Daniel Martin, who've done huge amounts of work and very different work in the area, come and listen to them. And then we'll also spend some time looking at the key points for submission. So it's just an hour and a half. There'll be drinks and nibbles around it, or coffee and tea and nibbles around it. So please join us. Please share that. And also we'll do a Zoom. There's Zoom as well. But if you want to be on Zoom, you need to see the UBC email and send us a note, and we'll send you the link.
Perfect. Thanks, Kolma, for that.
Thanks, Kolma.
Okay, no worries. Well, I think if there are no more questions, we might wrap tonight's webinar up. So thank you so much for agreeing to come on and do this, our first webinar, Hans. We really, really appreciate it. And we hope we can see everyone at our next one next month. And thank you everyone for coming.
My pleasure. Thank you.
Perfect. Thank you, everyone.
Cheers.